Okay, welcome to the Nearpod lesson for motion perception. So this is going to pick up where we left off, where we will leave off on Thursday in class. So we're going to talk about in this Nearpod lesson, we're going to talk about eye movements and what role they play in motion perception. And then we are going to talk about how the brain processes motion. And lastly, we are going to talk about biological motion. Okay, so in class, we would have talked about a couple of different types of eye movements, so smooth pursuit eye movements. So we're picking up with saccades. So saccades are another type of eye movement that we make. They are eye movements that we make when the eyes rapidly change our point of fixation from one object or one location in the visual scene to another. So saccades, they are very rapid. We can make them voluntarily or involuntarily. So we can make them on purpose or not. Let's look at what saccades look like. So if you were to measure saccades, you could do so using something called an eye tracker. So an eye tracker, it's like a camera that is mounted on your face and it records the movements of the eye. So it can trace your point of fixation as you look at an image. So this figure that I have here on the slide, this is showing the changes in the direction of a person's gaze while they are looking at this photograph. So let's talk about what these different, because this looks pretty messy. So what's going on here? Okay, so we've got this statue of a face. So the thin straight lines that we see, so thin straight lines, so like from here to here, this thin straight line, this thin straight line, this thin straight line, these represent saccades. So they're quick ballistic eye movements that we use to align the fovea with particular parts of the visual scene. So why do we say they're ballistic? Basically, saccades are referred to as being ballistic because once you start making them, you are not going to stop that eye movement. So if you're shifting your gaze around to different parts of an object, you can't start to make a saccade and then stop halfway through. So that's what they mean by their ballistic eye movements. You can't interrupt them once they are in progress. So the thin straight lines, these represent saccades. The denser spots along these lines, so you can see, oh wait, I don't have my cursor. Okay, so our thin straight lines are these ones here. And then we've got these dense regions here. These denser spots along these straight lines, these represent the points of fixation where we would stop, pause for a period of time to take in the visual information from that area of interest. So with, you can see here, in this image that there's certain parts of the object that we tend to fixate on, right? So we make ballistic eye movements from, one part of the scene that we're interested in to another. So say we fixate a lot on the eyes, right? And the nose, kind of the profile of the face, and then maybe we jump over here to the ear to focus on that for a while. So these show you the points of fixation. So when we stop making those saccades to kind of focus on that particular part of the scene. So an interesting thing happens while we are kind of in flight making a saccade. So when we make a saccade and our eyes are kind of in flight, the visual system essentially shuts down for the duration of the eye movement. So this is called saccadic suppression. So this refers to this reduction in visual sensitivity that happens when we make saccadic eye movements. So this just means that our visual system briefly shuts down. It's suppressed while our eyes are in flight. So the visual system doesn't shut down altogether. What saccadic suppression does is it suppresses information carried by the magnocellular pathway, which is the pathway to the magnocellular layers in the lateral geniculate nucleus, which are the layers of the lateral geniculate nucleus that process motion. So remember, we had the parvocellular layers that are more responsible for seeing color, fine detail. So the magnocellular layers of the LGN, they are responsible for processing motion, among other, kind of less fine detailed parts of the image. So saccadic suppression suppresses information carried through the magnocellular pathway, which is the part of the LGN that processes motion. So the visual system suppresses vision during saccades, and this is a good thing because it prevents us from perceiving motion when there's not any there. So saccadic suppression eliminates what would be the smearing of the retinal image that would happen if our eyes if we were taking in visual input while our eyes were in flight. The image would be quite smeared. So saccadic suppression prevents us from perceiving motion when there is not any there. Okay, so our visual system does shut down to some degree, at least in terms of processing motion when our eyes are in flight. So this brings us to the question of how do we discriminate motion across the retina that is due to eye movements versus that which is due to movements of objects? So how do we tease apart information that comes from our eye movements? How do we separate that from the motion of objects? Because both of these scenarios create movement on the retina. So how do we distinguish between our eyes moving and objects moving? Because both of these scenarios creates movement across the retina. So one explanation for this is called the corollary discharge theory. So let's look at this, how this works. So what happens when we make an eye movement? So what happens when we make an eye movement is that the brain sends two copies of that motor command. So we're going to make an eye movement. We're going to make an eye movement, and the brain is going to send two copies of that motor command, one copy to the eyes and one copy to something called the comparator. So let's just look at what these signals are first. Okay, so we've got our signal coming from the motor cortex. So that is going to bring a signal to the eye muscles to tell them to move. Now, split from this signal is the corollary discharge signal. This gets sent to the comparator, which we'll talk about in a minute. So the idea of the comparator is basically, it's a theoretical construct that compares the image motion signal with the eye motion signal. And this allows the brain to compensate for changes in the image that are produced by the eye movement. So, so kind of by sending out two copies of each order, so the motor signal and then the copy of it, the motor system can then, kind of solve this problem of, whether the eyes are moving or the object is moving. So let's look at the comparator next. So the comparator, like I said, it's a theoretical construct. It's a theoretical area of the visual system that receives one copy of the movement order issued by the motor system when the eyes move. The other copy goes to the eye muscles. So the role of the comparator is to compensate for the image changes that happen on the retina that are caused by the eye movement itself. So yes, the comparator, it is a theoretical construct, but it is believed to be the superior colliculus. So the superior colliculus is an important structure located in the midbrain. So this just gives you a couple of visuals. So we've got a side visual. So this is as if we had split the hemispheres down the middle, and we're looking kind of in the internal capsule. So this would be roughly where the superior colliculus is, and then if we kind of took this, took this region here and swung it around frontwards, this is where the superior colliculus would be. So it's tucked right in there. So the superior colliculus, it sends information to the eyes telling them to move. And the superior colliculus also sends a copy of this motor command so it sends it to the eye muscles and to subcortical regions of the brain as well as brainstem regions, so that the movements can be compared. So the signals can be compared. So the brain already knows ahead of time that an eye movement is coming. It doesn't have to wait for information coming through the optic nerve from the retina. So through the superior colliculus, and with this idea of the superior colliculus being the comparator, this essentially gives the brain the inside scoop on the upcoming movement ahead of time. So the corollary discharge signal, that is the term for this copy of the motor command that I had mentioned a couple of slides ago. So the corollary discharge signal influences sensory information processing around the time of the movement. So corollary discharge signals are important for identifying sensory input that happens from self-motion, so that things like the timing, the strength, the direction of self-generated movements can be compensated for. So this means that the nervous system can use that corollary discharge signal as a flag to identify sensory signals arising from self-motion and account for these signals when we're processing and evaluating signals coming in from the environment. So the corollary discharge signal, it's a copy of the motor command that's sent to the eye muscles to produce an eye movement. It's an internal copy of an upcoming movement that is sent to the brain regions that are going to be involved in that upcoming movement. So you can think of it like an internal memo that keeps everyone informed as to what's going on. In this case, everyone means the brain regions involved in the movement. So the copy of the corollary discharge signal does not produce any movement itself, but instead is directed to other regions of the brain to inform them of the upcoming movement. So this internal copy is theorized to contain the motor commands of the movement and what the predicted sensations of the movement will be. So this prediction enables our brains to estimate the sensory feedback that would be expected from the movements, which means that we can make corrections and adjustments to our movements in real time instead of having to wait for the brain to receive the information after the movement has already occurred. So it gives an estimate of the sensory feedback from the movements, and this allows us to make predictions about sensory outcomes. So not only does this corollary discharge signal, help us know if objects are moving or if our eyes are moving, it also helps us it helps us use sensory feedback to guide our movements kind of online instead of having to wait for sensory feedback. So it allows us to estimate the sensory feedback from movements, make predictions about what those sensory outcomes will be, so that we can make adjustments for our movements online. Now, interestingly, this is why you can't tickle yourself. So first, though, this internal copy of the movement is sometimes referred to as the afference copy. So the corollary discharge signal, the afference copy, means the same thing. So, the afference copy, the corollary discharge signal, and the fact that it gives us that prediction of the sensory outcomes of our movements, this is why we can't tickle ourselves, because the signal, if you're trying to tickle yourself, the signal predicts your own movement and effectively cancels out the tickling sensation. Now, that's not to say you can't tickle yourself. Certainly you can, but it's not as effective, it's not as ticklish as if somebody else does it, and it's because of this afference copy, this copy of the motor command that we get. So corollary discharge theory tells us that movement perception depends on three signals. So we've got kind of three things going on to indicate that we can use to indicate where the movement is coming from. So one, we have the image movement signal. So this is the movement of the image across the retina, which is going to stimulate receptors in the retina. So the movement image signal, this is the movement of images across the retina. So that's one signal that we have. And we also have a motor signal. So this is the signal that's sent from motor cortex, frontal eye fields as well, that indicates send a signal that actually moves the eye. So this is the signal that initiates the movements of the eyes. And then, split from the motor signal, we've got the corollary discharge signal. So this is the that internal memo that is split from the motor signal and gets sent to the comparator. So also known as the superior colliculus. So we've got the image movement signal that's sent to the comparator because if we think about the visual pathways and the visual pathway going from the retina to the LGN to the visual cortex, remember that some of those signals from the LGN go to the superior colliculus. So most of the signals coming along the visual pathway from the retina to the LGN, most of them go to the visual cortex, but a small portion of those fibers leave the lateral geniculate nucleus and go to the superior colliculus. So this is how the superior colliculus, also known as a comparator, gets that image movement signal. Okay, so we've got the image movement signal, we've got the corollary discharge signal. So let's talk about the scenarios where we perceive movement based on these three signals, and when we don't perceive movement based on these three signals. Okay, so movement is perceived if the comparator receives input from the corollary discharge signal, which is that internal memo that tells our brain that we're producing a movement of our eyes ourselves. And if we also get that image displacement signal coming from the retina telling us that an object has moved across the retina. So if we get both of those signals, movement will be perceived. So movement will not be perceived if we get the corollary discharge signal and the image placement signal happening at the same time. So if the corollary discharge signal and the image displacement signal arrives at the comparator at the same time, we won't perceive movement because these signals will cancel each other out. So if the movement not perceived, if the comparator receives both a corollary discharge signal and an image displacement signal at the same time. So if these signals come in at the same time, they will cancel each other out. The brain will know that that image displacement signal was sent because our eyes moved. So this will not be counted as real movement from an object. So eyes are moving at the same time as the image displacement signal. That tells us that movement across the retina is the result of the eyes moving, not the result of an actual object moving. Okay, so movement is perceived if we get a corollary discharge signal and an image displacement signal. But if they're not happening at the same time, movement will be perceived. Now, movement not perceived if the comparator gets those signals at the same time. They cancel each other out, and we will know that any displacement across the retina is due to the eye movements themselves. Okay, let's look at some behavioral demonstration that illustrates the importance of that corollary discharge signal. So we can try this right now. You can try this on your own with your own visual system. So what I would like you to do is just close one eye, doesn't matter which eye. You just close one eye, and then with your index finger, reach up and gently press back on your lower eyelid of the eye that's opened. Okay, so you got one eye opened, you got one eye closed. The eye that's open, take your finger and gently press back and forth on your lower eyelid. Now, as you're doing this, you probably notice that your eye is jiggling a little bit as you press on it, and it will kind of seem like the world is jiggling around. So this isn't how you usually control your eye movements with your fingers, obviously. So in this case, there's no corollary discharge signal. There's no copy of the message being sent from your eye muscles because your eye muscles aren't moving your eyes. So, excuse me, since there's no corollary discharge signal to tell the visual system to subtract out the motion caused by this eye movement, you'll notice the very unusual perception as you're pressing on your eye that the whole world kind of seems to jiggle around. So as you move your eyeball up and down, the projection of the world on your retina shifts around. And if that happens without your eye moving, this would mean that the world has moved up and down. So if your eyes aren't moving, your eye muscles aren't getting those signals, this would tell you that the world is moving up and down. And that is what you perceive. In that demonstration, it looks like the world's moving around. So when we jiggle the eyeball with a finger, no signal is sent to the eye muscles, to the comparator, to the superior colliculus. The eye muscles are not what is moving the eye, so no signal is sent. So the visual input is interpreted as our world jiggling around because all that we have is an image displacement signal in this situation. We have no motor signal because you moved your eye with your finger. So this means we have no corollary discharge signal because there's no motor signal. So if we just have an image displacement signal, your brain assumes that the world must be producing the motion. Let's look at some physiological evidence for corollary discharge theory. So damage to a particular part of the temporal lobe that's important for motion, the medial superior temporal area. So when people with damage to the medial superior temporal area, when people have damage to this region, when their eyes move, they'll perceive movement in a stationary environment because there is no corollary discharge signal to counteract their own eye movements. So in non-human primates, there's also evidence for real movement neurons. And these real movement neurons are neurons found in the medial temporal area that respond only when a stimulus moves. So if we look at this, so we've got our monkey. It's watching a bar move across its visual field. So as the bar moves across the visual field, we get an increase in firing rate for these. So in this situation, the monkey would be having an electrode implanted into the medial temporal area, which is involved in motion perception, while this bar moves across the screen, and then we would get patterns of activity such as this. So this is the bar. It's moving across the receptive field of this neuron that we are recording from. And as the bar moves across the receptive field, we get this increase in firing associated with the movement of the bar. Now, on the other hand, so these real movement neurons, if on the other hand, this monkey is making an eye movement at the same time as this bar is passing through across the receptive field, you can see here that that peak in activity has in these real movement neurons is significantly less. So these real movement neurons respond only when a stimulus moves and not when our eyes are moving. So if our eyes are moving and an image displacement signal is being sent that this bar is moving across the receptive fields, again, the brain knows that the eyes are moving, so we don't get this increase in activity in these real movement neurons because the brain knows this isn't real movement, it's our eyes moving. So the eyes aren't moving, the eyes are stationary, and the bar moves across the receptive field. We get increased activity in these real movement neurons. If we're making an eye movement while the stimulus moves, we don't get this increase in activity in these real movement neurons because the brain knows the movement is due to our eyes. So corollary discharge theory, we also have evidence that that the brain does compensate for eye movements by remapping the receptive fields of neurons in the posterior parietal cortex and the frontal eye fields. So, so these are the regions, important for making eye movements. So we've got frontal eye fields, we've got the supplementary eye fields, and we've got the parietal eye fields. So, this is one way that this compensation for eye movements happens. It's through this remapping of visual fields. So, we know a lot about visual fields and how they work from previous chapters. So the visual receptive fields, this is just regions of space where a visual stimulus causes a neuron to change its firing when something enters that area. So single cell recordings from non-human primates have shown that the receptive fields of some neurons in the parietal cortex can change in advance of an upcoming eye movement. So, for example, if a saccade is planned, so one of those rapid ballistic eye movements is planned but not yet executed. This is one type of experimental task that can show this remapping of receptive fields. So, so let's just look at this here. So, if we have, so we're showing the monkey these picture of an apple, picture of a pepper. So this is where they're currently looking, right? So their fixation, this is where they're currently looking. So they're currently looking at the apple. And this is the activity in this receptive field right at the moment when the monkey is looking at the apple. So the blue lines represent the activity associated with the apple, and this kind of magenta represents the future receptive field. So basically, the monkey knows that the task right now is to look here, and then something will come on the screen and tell them that this is going to be the future eye movement that they're going to make. So they're looking here, and then something will come on the screen to tell them the next eye movement that you're going to make is going to be to the pepper. And then if you then measure the activity of associated with this receptive field now, right before the saccade is going to happen, you see activity then associated with the future receptive field. So these neurons in the parietal cortex and frontal eye fields, they remap the receptive fields depending on the upcoming location of the fixation. So information about the corollary discharge signal is important, and it's actually more useful than the information that we get from the eye muscles. And this is because the corollary discharge signal is sent before the eyes move, whereas the signal sent from the eye muscles, this happens during the actual movement. So because the signal from the eyes is sent during the movement, the brain would only receive this information later on after the movement is over. So, so the corollary discharge signal gives us better information because it is sent before the eyes move. So, and also combined with the remapping of those receptive fields for neurons in the parietal cortex, the fact that they change their receptive field location just before an eye movement happens. So we have neurons in the parietal cortex that seem to know what the eyes are going to do before the movement happens. And they're prepared for the movement in a way that suggests that they have some predictive information that is not coming from the eye movements themselves, because those eye movements have not happened yet. So if I flip back here, this scenario here, no eye movement has happened here. No eye movement has happened. They're still fixating here. But because they know that the next saccade target is going to be over here, you get those neurons responding to this location before the eye movement has been made. Okay, so next we're going to look at a video that explains something referred to as Reichardt detectors. So Reichardt detectors are neurons that fire, they're theoretical neurons that fire to movement in one direction or the other. So we're going to watch a video here, and then we're going to start talking about motion, perception, and how it is processed in the brain. Okay, motion, perception, and the brain. So when those signals leave the retina, they travel along the magnocellular pathway when we're talking about motion. So the magnocellular pathway in the lateral geniculate nucleus carries that low spatial and high temporal frequency information that's associated with motion. So we're talking about those coarse motion details things moving out of the corner of your eye that catch your attention. These are coarse motion details. So the magnocellular pathway carries this low spatial quality but high temporal frequency information. And carries it to regions of the middle temporal area, MT, the middle superior temporal area, MST. We'll talk about those regions in a bit. Those are the cortical regions involved in motion perception. And they are, okay, so we've got our visual pathway coming through the magnocellular pathway of the LGN, going along to the primary visual cortex, and then those signals start to travel along the dorsal pathway. So the medial temporal lobe and the middle superior temporal lobe, these regions here are parts of the dorsal pathway, and they're important for motion perception. So the magnocellular pathway carries information about, large, fast-moving things. So low spatial frequency, high temporal frequency, broad strokes movement. Whereas the parvocellular pathway carries that information about small, fine detail, slowly moving, colorful things. So one way that we know that the magnocellular layers of the LGN are important for motion perception is that if lesions to this pathway are created either experimentally or in rare cases of brain damage in humans, this will impair perception of large, rapidly moving objects. So the first port of call for visual information in the cortex is the primary visual cortex, whether we're talking about, motion or other perceptual qualities. So perception of motion begins first in the striate cortex, the primary visual cortex. So V1 neurons do respond best to oriented lines, as we know, and some of these neurons are also selected for the direction of light or dark edges against a light or dark background. So when such a stimulus is moved across the receptive field of V1 neurons, light dark edges against a light or dark background, V1 neurons will respond to this. So this would be, these would be considered your complex neurons. So if you think back to the different types of V1 neurons we talked about, these would be our complex neurons responding to motion across the receptive field. So the neural architecture, if you will, that is necessary to perceive motion is already in place at the relatively low level of the visual system, that being V1. So we do have some motion perception abilities as low as V1. But there is a problem with these individual motion detectors that are found in V1. So this problem with individual motion detectors in V1 is referred to as the aperture problem. So the problem with the individual motion detectors in V1 is that the direction of oriented lines or bars moving through the receptive fields of V1 is pretty ambiguous. So you can think about the type of what V1 neurons are getting is kind of like what a camera, it's kind of like they're getting snapshots snapshots of the movement, because V1 neurons, they only see a small portion of the world based on their receptive field and where it's directed at. So they don't respond, they don't respond, maximally to, a single stimulus, form a single oriented bar, but to kind of a whole family of motions. So we have to integrate the outputs of many of these motion detectors in V1 over larger and larger portions of the visual field. And it's difficult for the visual system to, clarify these signals and be able to determine if genuine motion, is happening, because the signals that the individual cells in V1 get are inherently ambiguous. until later stages in processing when we can extract global motion. So global motion is occurs, or global motion is extracted at later higher visual regions such as MT. But first, let's kind of look at a demonstration of the aperture problem, and then we will move on to how this problem of ambiguous motion is solved. So just give me 1 minute. So what happens with the aperture problem in this demonstration is that when these striped patterns are seen through these circular apertures, like they were in that first video, like right here, they appear to be all moving in the same direction, right? They appear to be moving up and to the left. But if we expand that visible region, we can see the true motion of the striped rectangles behind these circular apertures. Now the stripes within each of these three circles appear to be moving in different directions. So why is there such a dramatic change? Well, when the three rectangles are visible, their edges provide unambiguous motion cues that help our visual system interpret the true motion of the stripes within those circular apertures. Without this global motion cue that those edges give us, our visual system just finds the simplest explanation for that local motion within those circular apertures. And the simplest explanation is that they are all moving up and to the left. So when we don't have those global motion cues to tell us otherwise, the simplest explanation is that moving up and to the left. So this is the type of thing. You can kind of think of these apertures as representing, what each what say 3 particular cells in V1 would get. So it ends up being ambiguous until we have these global motion cues, which are contributed from regions outside of V1. So the aperture problem shows us that we need global motion cues to help our visual system resolve the ambiguous motion signals that we get from V1 neurons. So when we're viewing only a small portion of a larger stimulus, this can result in misleading information about the direction in which that stimulus is moving. So that's what we saw with the circle apertures. And this is analogous to the activity of a single neuron in V1, a complex neuron, but it doesn't provide accurate information about the direction of the movement. So we need to integrate the outputs of many motion detectors over larger and larger portions of the visual field in order to, make these signals less ambiguous so that we can extract genuine motion from what really is an inherently ambiguous local motion signal that we're getting from V1 neurons. So this, we need to extract global motion cues. So where are these global motion cues extracted from? So where are the local motion cues integrated into global motion cues? Such a process is thought to occur in a region called MT, the medial temporal area, and the medial superior temporal area, MST. So both of these regions contain cells that respond to coherent global motion. So Hubel and Wiesel, who we looked at earlier when we were talking about receptive fields of V1 neurons, they also discovered direction selective neurons in the cat, and they found that almost all of these direction selective cells were found in the medial temporal area. So most of the cells in MT are indeed direction selective. So the middle temporal area plays an important role in motion perception. The vast majority of neurons in the medial temporal area are selective for motion in a particular direction. So just like the neurons in V1 that we have that have a preferred orientation, they have a preferred eye, neurons in the medial temporal area have a preferred motion direction. And as you can imagine, there are also populations of neurons grouped together that have a shared preference for a particular motion direction. So it's a similar type of thing as when we were looking at, orientation preference in V1. So we're going to have groups of neurons in the medial temporal area that are grouped together, that respond to similar directions of motion. So some studies have looked at this, and so how do you measure motion movement? So a couple of researchers, Newsom and colleagues, they looked at the responses of these neurons in the medial temporal area to motion coherence. So motion coherence, basically just they had dots moving in a direction that was either, so they would have all of dots moving in one direction across the screen. So this would be 100% coherence. So 100% correlation means that 100% of the dots are moving all in the same direction. So all the dots are moving in the same direction. And then you can have all the way down to no correlation and zero coherence. So this is when the dots are just moving randomly. You can't distinguish any pattern of motion because there isn't one. And then you can have everything in between, right? You can have 50% coherence, where half the dots are moving in one direction and the other half are moving in the other direction. You can have 25% coherence. You can have 75% coherence. So they took the coherence of these, the movement coherence of these dot patterns, and they varied it. So they varied it, and then the monkeys would go through a training period where they were taught to judge the direction of these dot movements. So they would, get pretty good at this task. So they would be shown all these different movement coherences, and they would be able to push a button to indicate, the direction of the dot movements. So once they learned how to do this task, once they learned how to, indicate the motion, the direction of motion from these dot displays, they were then, suited with recording electrodes in the medial temporal area, so that the responses of the neurons in the medial temporal area could be measured while they were making these direction judgments. So what they found, Newsom and colleagues, they found that neurons in the medial temporal area did respond to motion coherence. And their results showed that as the coherence of the dot movements increased, the firing of neurons in the medial temporal area also increased. So basically, the more coherence that was in the dot display, the more firing they would see in the medial temporal area neurons. So if you have 75% coherence, you would have more activity in MT neurons compared to 25% coherence. You would have less activity in these MT neurons. So the accuracy of the movement, so as the coherence of the moving dots increased, the firing of these medial temporal neurons increased, and monkeys were also more accurate with judging the movement direction. So they also looked at this another way, Newsom and Perret this time. So they looked at lesioning the medial temporal area to see what that would do to the monkey's ability to detect motion. So they did a lesioning experiment. So they found that, first, if you have a regular monkey, so a neurologically intact monkey that has been trained on this dot motion detection task, these monkeys can be very good at this task. They can detect motion with a coherence of only 1 or 2%. So having only 1 or 2% of the dots moving in the same direction, all the other ones moving randomly, they can actually detect 1 to 2% coherence of motion. That's pretty good. But monkeys with lesions to the medial temporal area, on the other hand, their ability to detect motion coherence drops down significantly. And they need to have motion coherence of at least 10 to 20% in order to correctly detect the direction of the motion. So a neurologically intact monkey that doesn't have damage to the medial temporal region can detect motion that has only a 1 to 2% coherence. Once these monkeys that were once good at detecting this low level of coherence, once they have lesions to the medial temporal area, all of a sudden they can't detect motion until that coherence is at least 10 to 20%. Okay, what about looking at this another way? So we've got, recording from neurons in MT. We've got lesioning MT. What about stimulating the medial temporal area? So this has been done as well. These in these studies, the monkeys, they'd be looking at dots, moving a certain direction, and they would have to, indicate. So in this case, the dots are moving to the right. So they would press a button to indicate that they know the dots are moving to the right. So this is the dot movement. This is their perception of the movement of the dots as indicated by their response. So in this case, the dots are moving to the right, and their perception and their response is that the dots are moving to the right. Now, if they stimulate it, so Britton and colleagues, if they were to stimulate neurons in the medial temporal area that have a preference for downward motion, this is what happens. So the dots are still moving to the right. They stimulate they stimulate neurons in the medial temporal area that have a preference for downward motion. And then all of a sudden, the monkey has responds that the dots are moving down and to the right. So their perception is altered by that stimulation, and what happens really is that that it's creating a bias in their perception, a bias that is in the direction of the preferred motion direction for those MT neurons. So these MT neurons that they're stimulating have a preferred down direction. This changes their perception. It biases their perception in the direction of the stimulated neurons. So neurons in the medial temporal area, this is where a global motion is extracted, and a lot of evidence showing through recordings, through lesioning and through stimulation that, that there are neurons in the medial temporal area that have a preferred motion direction. And if you stimulate neurons with a preferred motion direction, it will bias the perception of that movement in the direction of the stimulated neurons and their preference. Okay, the last topic that we're going to look at in this Neuropod lesson is biological motion. So we are highly sensitive to motion in the peripheral regions of the retina. And this is good because this is an important source for biological motion. So greater sensitivity to motion in peripheral regions of the retina may not be good for helping us see things like the rotating snake's illusion, which we talked about in class, or we will talk about in class. So having high sensitivity to motion in peripheral regions, not good for that type of illusion, but it is really good for our next topic, that topic of biological motion. This is more important than illusions, really, because illusions like the rotating snake's illusion, these are really just illusions that exploit things about our visual system that are actually really adaptive. So this is one good example, of that. Our visual systems, we are highly sensitive to motion in the peripheral regions of the retina. We're highly sensitive to it, but as we know, we don't get high visual acuity from these regions. So it's not good detail, but if we're talking about biological motion, This is a good thing to get this high sensitivity to motion in peripheral regions. So illusions are really interesting, but when it comes to our evolutionary history, we're not really going to be faced, or we haven't been faced with, images that are specifically designed to exploit the aspects of our visual system that make us susceptible to these motion illusions. So we would never encounter these situations, right, in an evolutionary sense. But biological motion is really important for survival. It's important for survival, and our oversensitivity to motion in the peripheral regions of the retina is super important for perceiving biological motion. Why? Well, because if we're really sensitive to motion in peripheral regions of the retina, we can just quickly move our fovea to regions of the visual field when motion occurs there so we can get a better look at it. So we're really good at determining biological motion, even if it is really stripped down to just dots positioned on theoretical parts of the body. So let's try this demonstration and see how many of these you can guess. Okay, so you probably surprised yourself on, how well you were able to distinguish the movements being carried out by these theoretical point light displays. So we are pretty good at determining motion from these really, these really stripped down displays, but our ability to determine what we're looking at definitely improves as more dots are added. The key thing seems to be that the joints are highlighted. So points on the joints improve perception of biological motion. So having, those light points on, on the feet, on the knees, on the hips, on the shoulders, on the waist, on the elbows. So having the points on the joints, this improves perception of biological motion. And another important factor is that there is coordination between the different points. So this is the most important, that the coordination between those points is appropriate. So we are good at biological motion perception. And it's not surprising that our perception of biological motion, we have a region for it. So the region that we have that's important for perception of biological motion is the superior temporal sulcus, and also the fusiform face area. Okay, so the perception of biological motion depends on a couple of key brain areas, one being the superior temporal sulcus, which is I have highlighted right here on the screen, and also the fusiform face area or the fusiform gyrus, which of course we know is important for perception of faces. So one way that we know that the superior temporal sulcus is important for biological motion perception is that if you apply TMS, transcranial magnetic stimulation, to the superior temporal sulcus, it interferes with our ability to detect biological motion. So if you apply pulses of TMS that are delivered in such a way that they disrupt functioning of the superior temporal sulcus, our ability to detect biological motion will not be as good, so it will decrease. So biological motion is important for a variety of reasons. One reason is that biological motion plays an important role in how we understand the actions of others. And we are also, it's important for, navigating our complex and dynamic social worlds. So having that ability to, perceive, interpret, and respond appropriately to social cues, which are displayed through body language. Our perceptual systems are specialized to detect and attend to the movements of animate beings. So attending to that social information that we can get from others. Okay, let's do a recap of this chapter. So in class, we talked about the different problems that we encounter when perceiving motion. We looked at scenarios where we perceive real motion and scenarios where we perceive motion that's not there. So we looked at what the mechanisms are for real and illusory motion perception. And then starting in the Nearpod lesson right now, we were looking at what eye movements contribute to motion perception. So smooth pursuit eye movements, saccades, the corollary discharge signal. So how, our eye movements are important for motion perception, but at the same time, they can also interfere with it if we don't have, mechanisms in our brain to compensate for our own movement so that we can distinguish, our own movement from movement of the world. So eye movements play an important role in motion perception, but we also have to account for them in, we have to account for them so that we can factor them out to prevent us from perceiving motion that isn't there. So one way that this is done is through the corollary discharge signal. And then right now we just talked about also biological motion perception. Okay, guys, so that's it. You'll do a little bit of a quiz here at the end. And this wraps up the chapter for motion perception.